say out of the blood in the mouse model, out of the blood and into the lung, or in the tissue culture, whatever motility assay we're using. So what do they do to move that's different from the non-invasive ones and the non-modal ones?
Well,
you could say you don't know. I mean, I don't know.
Well, I'm just trying to understand your question, because if you ask me about things that the cells require for movement, so they require, for example, actin remodeling, I don't think there's anything known about p63 acting on actin remodeling. However, you could argue that if it controls ERK, then ERK can affect actin remodeling. So, but that's just an idea. Since p63 affects adhesive molecules, then changing the kind of adhesive molecules that you have, that affects whether cells can stick to other things or not, right? So if you change, for example, expression of E-cadherins or N-cadherins, then you can adhere to, say, mesenchymal cells instead of epithelial cells, and then they can just like pull you out of there, bring you into the mesenchyme, right? p63 has been reported to regulate N-cadherin expression, and when you get rid of it, then you see more N-cadherin. But then there are reports that say that it doesn't regulate N-cadherin. So the role of p63 on EMT in particular is not very well established because there are conflicting results. We have not seen changes on N-cadherin or on E-cadherin upon p63 overexpression or knockdown. So we think that it is by a different way, not through EMT, that the cells become more invasive. And that's my really roundabout way of saying that I don't really know. Yeah, Mike.
So you said that the delta N p63 seems to have like a different phenotype from the TA p63. What about having a transactivating domain present causes that change, or is it something else that what is that domain interacting with that might be causing this?
The TA domain?
Yeah.
So the question is, I believe, why are delta N p63 and TA?
So it seems like they have similar sub-isoforms as far as the other domains aside from the core domains. It seems like they have similar C termini, or at least, you know, the repository of the same ones. What about the TA domain that's different?
Well, the TA domain is homologous to the TA domain of p53.
Right.
So TA p63 isoforms can upregulate some of the same targets as p53, right? So, and that's why they can direct cell cycle arrest or apoptosis, right? TA p63 upregulates p21 expression, for example, which is for which it requires the TA domain, right? Because it's like p53. Delta N p63 inhibits p21 expression in some form, in some cell lines, not in every cell line, because it binds to the p21 promoter, but then cannot transactivate gene expression, right? With regards to the C termini, there's very little known about the differences between the different isoforms. It's really hard to study, especially because only the alpha isoforms are really expressed at levels high enough that you can do anything with them. So to answer your question, the main difference between the TA and the delta N with regards to the N terminals is that the TA isoforms are able to transactivate gene expression in a manner similar to p53 because of a p53-like transactivation domain. Steve.
Well, first, I'd like to congratulate you, congratulate Jim, because as a second reader, I think this is one of the best theses I've read at that stage. I practically had to do anything to change it. It was well written, really nice use of language that you don't normally see, in a lot of theses. And I think that students in the present and the future should take a look at your thesis and learn by learn by it. So congratulations. But now I have a question, though.
Yeah.
Can you go back to the previous slide? Based on this model, do you think there are other targets for MKP3? Because what came before this was the use of a powerful oncogene to activate ERK, as you said, you know. And it appeared when you knocked down P63, there was no dramatic change in ERK activity, you know. So is it conceivable that MKP3 is going to cell evasion via some other route?
Thus far, okay, there's one report in the literature that says that MKP3 has a different target. It's a FOXO gene protein. But more or less the consensus in the literature is that MKP3 has an exceptional affinity for ERK1 and ERK2, and basically insignificant affinity for, say, ERK5, which is the most similar ERK family member, and no affinity for JUNK or P38 MAP kinases. So there are no other targets that have been described in the literature. You know? So of course there's a possibility, but there's no evidence to suggest that.
If you treat mice with a U compound, they'll probably die, won't they? I was just thinking of some way to attenuate ERK in those mice after the knockdown to see whether ERK is the actual target in the mice of MKP3. You could do it by other means. You could potentially create a conditional cell where you conditionally knock it down after you've delivered it in the context of the mice. That could be done with any of the players. It's to sort of trace the metastatic event through by conditionally hunting down each of the regulators. Anyway.
We will get the students in China to do that.
Well, okay, any other questions? Okay, so we're going to have a party at 5 if he passes. Party in the lounge. Okay, everybody work. Okay, thank you for coming.